Hello, everyone, and welcome. Thank you for joining us today. My name is Kübra, and I'm a global marketing specialist from IVIM Technology. Today's presentation will be about the latest technology in live animal imaging, real-time intravital microscopy for biomedical research. It will be presented by Dr. Selina Soyeon, and during the webinar, if you have any questions, please use the Q&A box below. We will also answer some of the questions sent prior to the webinar. Selina, you may start with your presentation.
Thanks, Kubra. Hi, I'm Selina. I'm an applicational specialist in IVIM technology. So it's my great pleasure to meet you, even if it's virtual, but very happy to introduce our technology to you today. So for today's presentation, I'll briefly introduce our technology and the research applications of various disease animal models in preclinical research in vivo using IVIM system. So during webinar, if you have any question, please text it in the Q&A box. Then I will respond to your question on Q&A session as soon as possible. Firstly, I'd like to introduce IVIM's intravital microscopy to observe live tissue and cells in live animals. So compared to a conventional macro-scale imaging technology such as X-ray, CT, and MRI, which only provides the tissue level and static in vivo information. By the way, the intravital microscopy can directly observe the dynamic cell movements inside of the internal organ of the live animal model for the human disease. So this example video shows how red neutrophils inside of the vessel can form a cluster and block the green blood circulation in the lung of sepsis animal model. So thereby, intravital microscopy can provide a detailed live cell video of the disease development in cell level. Especially, we developed a specialized device for the lung imaging, which is the suction-based system optimized for in vivo lung imaging. So we've exposed the lung tissue through the lip incision, and then with the little negative pressure, which are applied through the suction hole of the lung imaging window chamber, then we can let the lung tissue can be attached to the cover slip in a stable manner. So with this in vivo stabilized lung imaging system, we can acquire the high resolution microscopic images of the neutrophil dynamics in the pulmonary vasculature in the live mouse model. Also, we can apply this principle to the other organs, such as the heart. So this is the vacuum-based in vivo stabilized heart imaging chamber. So with this system application, we can acquire the real-time video of the heart beating, visualize the red blood cell flow inside of the blood circulation in the heart. Sorry. And in the magnified view, we can see these flowing red blood cell dynamics inside of the blood vessel of the heart. Actually, the heart is beating actively, but we've just stabilized the small portion of the heart. Then we can acquire this real-time video in the cell level. So we can monitor and track the flow of the individual cell, single red blood cell, and also lysin positive neutrophils inside of the heart tissue in the live animal model. And not only the cell, but also we can image the drug actually. So this is the 24-hour image obtained from the cancer implanted mouse model. So in this data, the GFP expressing signal is the human breast cancer cell we've implanted to the dorsal skin of the dorsal skin fold chamber implanted model. And the cancers have just grown to a certain size of the tumor after the 2 weeks of the implantation. So then we treated the red fluorescent nanoparticle drugs to analyze the drug delivery and the drug distribution over the target breast cancer. So from 2 hours to 6 hours after drug treatment by IV injection, the red nanoparticle drugs firstly appear mainly inside of the vessel. And then finally, after 24 hours, we can see that the amount of nanoparticle drug just get out of the vessel and finally delivered to the cancer tissue. So this long-term monitoring over 24 hours successfully revealed that the nanoparticle delivery to the tumor through the vasculature and the uniform distribution in the target tumor tissue. So this is one of the very good example how we can utilize in intravital microscopy technology to monitor the drug delivery to the target cell or the target tissue or the organ inside of the living animal. So conventionally for in vivo experiments, users have to prepare each required functions by themselves. There was a high degree of the difficulty and non-optimized performance with limited application to the imaging interest organs. It was very difficult and it's not an optimal solution with the low performance without any standardized in vivo protocols. So IVIM's all-in-one intravital confocal and two-photon microscopy system is extensively optimized and carefully engineered to provide the best performance inside of in the in vivo imaging of the live animal model. So IVIM's intravital microscopy enables 3D cell level microscopic imaging and analysis in vivo. So it successfully achieves the dynamic in vivo live cell imaging, such as real-time cell trafficking, cell-to-cell interaction, and the cell-to-microenvironment interaction inside of the living body. And furthermore, it can be widely applied to the therapeutic research field, investigating the drug delivery to the target tissue, drug efficacy, and therapeutic mode of action in various preclinical mouse models of the human disease. So using the IVIM system, it can be applied to the various research fields, including the basic biology research and some therapeutic research fields as well. So here are the applications to various preclinical research fields, such as in vivo analysis of the immune response, tumor model, and the disease model. And for the drug development processes, we are validating the drug distribution in the target liver organ, drug delivery to the tumor tissue, and also we can validate some drug efficacy inside of the skin and the tumor as well. So IVIM is the world's first and only one company providing this new all-in-one system with the best in vivo imaging performance. This all-in-one single box package enables easy installation, operation, and maintenance, and it has co-optimized hardware and the software for the best in vivo imaging performance with ultra-fast imaging speed, which is up to 100 frames per second. Thereby, we can even capture the fast dynamics, such as red blood cells that are flowing inside of the blood vessels. And also together with the automatic hassle-free motion compensation function and the various accessories such as live animal maintenance system and the various imaging window system for the wide range in vivo applications, users can easily get the high quality in vivo data without any limitation in the imaging various live organs. So our first key feature is the ultra-fast imaging speed. So all of our all-in-one intravital microscopy system is fully equipped with ultra-fast laser scanner, which is polygon scanning system. It enables the ultra-fast imaging speed in vivo at the maximum speed of the 100 frames per second with 512 by 512 pixels. And also it could achieve the uniform excitation illumination over the entire imaging field of view. Thereby, we can acquire the high quality images and the videos at ultra fast frame rate in real time. And this is very important to obtain the high quality in vivo data from the live animal by compensating the tissue motion artifacts as well. Otherwise, the other competitors usually have galvanoscanning resonance system, which only get the ununiform excitation due to the unstable scanning speed of the resonance scanner over the imaging field, and also with the limited scanning speed as well. So this is the polygonal mirror set, which are assembled in every product lines of IVIM. In addition to ultra-fast imaging, IVIM's intravital microscopy is fully integrated with the animal motion compensation function. It enables an automatic hassle-free, high-precision motion compensated images by facilitating the frame-by-frame motion compensation. This is one of the examples of the motion compensation function. This is in vivo real-time video of the heart imaging. So in this side-by-side comparison, even though the raw imaging data has the high degree of the movement, but when we apply the high quality of the clear image, apply the motion compensation function, then we can directly acquire the high quality of these clear images without any shifting artifacts. So without this motion compensation function, only this blurred image could be obtained due to the motion artifact, which highly disturbs the quantitative analysis of the cell dynamics of the live animal model. So I'd like to emphasize that the conventional standalone microscopy only can acquire those kind of shifted data, like in the middle, from the same live animal model. So this is the video actually working in the studio software. Just one click for the selecting reference channel for the compensation and activate the motion compensation button and click capture. Then we can directly acquire the compensated result in the display window right away. And we are also providing various additional accessories for maintaining the healthy condition of the live animal model during in vivo experiments. So here we have the rectal probe sensor for the body core temperature, which has a feedback system with the heating plate at the bottom. It fully maintains the stable body temperature throughout the whole experiment. And also there's a glass heater for the local tissue temperature maintenance of the target imaging organ or the tissue. Therefore, we can keep the stable status of not only the body core temperature itself, but also local tissue temperature of the imaging target organ simultaneously at once. So this is the translational stage for mounting the mouse model. So every accessories can be connected to the connector in the left. And we can slide in and out of the stage before and after mounting. And moreover, all of IVIM's intravital microscopy has its own operating and the post-processing software. The name is IVIM Engine and IVIM Studio. So with the IVIM Studio software, we can perform this kind of cross-sectional analysis of the cell nucleus and the vasculature in the villi of the small intestine, and also could identify the co-localization of each cells in 3D. The following is the 4D data we've acquired by the 3D imaging of the ear skin over time. The 4D analysis of the cell dynamics and the cross-sectional co-localization analysis can be available in real time using Studio software. And with the measurement function, the quantitative analysis of the vessel dilation before and after drug treatment, and also some immune cell trafficking or cell-to-cell distance and cell-to-cell contacts can be quantitative analyzed in the Studio software. In addition, for the quantitative analysis of the immune cell movements, cellular speed, direction, and cell-to-cell distance, and the number of contacts over time can be performed in 4D in the Studio software. In addition, we have developed the new image processing function in collaboration with the Professor Young-Kyu Yoon's lab in the Department of the Electrical Engineering at KAIST. So this function is an AI-based noise canceling feature for the real-time videos. So with this feature, the high-resolution live cell videos can be obtained in real time at very fast raw video speeds of 30 to 100 frames per second without additional averaging processing and also without additional frame loss, as you can see. As an example applied to the muscle, we can record the video of the RBC flow at high resolution and also real-time frame rate. And with this function, it enables quantitative analysis of the cell trafficking and the other cell dynamics with high resolution and the high spatial resolution as well. High temporal and high spatial resolution. And finally, this is the newly launched model this year, dual mode intravital confocal and two-photon microscopy, which is IVIM CMS3. It is the most compact and affordable in vivo confocal and two-photon microscopy in the market, providing the versatile functionality in the single box. With the confocal laser and the compact two-photon laser unit with one switch mode changing feature, IVIM CMS3 can provide the comfortable multipurpose use for in vivo functional imaging and also really saves from the unnecessary space and the high cost. This is the simple introduction video of the IVIM system, which has both confocal and multiphoton system in one single box, single device. So there's an animal stage where the live animal can be mounted on like this. And before imaging, we can select the objective lens. Then when we start the imaging, so yeah, this is the schematic movie of the laser beam path during the imaging. So this is the excitation of the confocal system and the emission beam path for the confocal. Compared to the confocal system, which has the 4 laser source, the two-photon system has only one source to excite the fluorescence. Then the emission signals can be separated into the 4 colors and detected by the 4 detectors each. Then by using motion compensation function, we can get the high quality in vivo data in real time. And through the post-processing software, the 4 color of the simultaneous 3D and 4D imaging analysis can be performed. Using IVIM CMS3, we can easily acquire the high-quality 4-color in vivo imaging of various tissues in the animal model. This is the example of the in vivo skin imaging visualizing the collagen in white color, single-cell nuclei in green color, cell bodies in red, and the blood vessel in blue in vivo. So for this, we've utilized the H2B-GFP and actin-DS-RAD MTMG double transgenic mouse, which express the GFP in each nuclei and express TD tomato in every cell body simultaneously. Then we did additional in vivo labeling of the blood lumen by IV injection of Evans blue. The color can be the blue color. Thereby, we could visualize the blood vessels in blue color. This is in vivo muscle imaging of the same mouse model. We can clearly visualize the detailed structure of the striated muscle tissue, which are composed of the sarcomeres, myofibrils, and also the vessels. In pancreas, in vivo visualization of acinar cell and the pancreatic islet cells can be acquired with the best imaging quality in vivo. In the kidney, the renal capsule expressing the high intensity of the second harmonic generation collagen structures, and the renal cortex and the glomerulus can be visualized in high resolution in vivo. In the spleen, there are also a lot of collagen signals expressing from the surface of the spleen. And when we see the more detail inside, a number of the various immune cells supposed to be the T cells or the B cells, and some splenocytes were visualized in green color, and also the blood endothelial cells were imaged in the red color, and blood lumen in blue color each. So from the IVIM system, users can do various preclinical research analyzing in vivo immune responses in the live animal model, tumor model, and also human disease animal models. Also, we can utilize in vivo microscopy imaging technology to monitor the drug delivery to the target cell or the tissue, or in the live animal model, as well as we can validate the drug distribution in the target liver organ, and can validate some in vivo drug efficacy in the skin and also the tumor. Finally, IVIM's intravital microscopy has succeeded in high quality live cell imaging in almost all internal organs in the live animal model. It includes some brain, even retina, and some GI tracts inside of the abdomen, prostate, lymph node, mammary tissue, liver, muscle, and even heart and lung. So from this slide, I'd like to introduce the research application in detail according to the target organ. So this is in vivo imaging of the ear skin, visualizing the immune cell dynamics in the skin of Lyme GFP transgenic mouse, together with the ear skin blood vessels. As we can see here, we could visualize some cell-to-cell dynamics of the neutrophil in the skin, and also can acquire the very fast cell dynamics of the flowing neutrophils through the blood flow. And here's the research example of the drug efficacy of the CARS protein, one of the secreted amino acid, tRNA synthetase. So to investigate the details of how this enzyme can induce inflammatory responses in vivo, we monitor the immune cell dynamics after intradermal injection of the CARS protein to the ear skin tissue. A time-lapse in vivo imaging showed the active recruitment of the macrophage and the monocytes to the injected KRS. Then it finally enables in vivo analysis of the inflammatory effects of the therapeutic material in vivo. For immunological research, we can directly monitor the immune cell dynamics in the popliteal lymph node. We've adaptively transport T cells and the B cells, which are obtained from the actin DS rat and the actin GFP transgenic mouse, respectively. And then we monitor the extravasation dynamics of the T and B cells in high endothelial venule of the lymph node in real time. So here, the T cells and the B cells are selectively extravasated through the specific spots of the high endothelial venule. And to monitor the stepwise transmigration process of the T cells in high endothelial venule, we perform the intravital 4D imaging analysis of the T cell migration across the high endothelial venule. It clearly shows that the multiple steps of the T cell transmigration across the high endothelial venule, which are composed of the endothelial adhesion, intraluminal crawling, trans-EC migration, intra-perivascular channel crawling, and finally getting out of the high endothelial venule, arriving at the lymph node parenchyma. And from the long-term time-lapse imaging, we could identify that there are some hotspots in high endothelial venule where the trans-endothelial cell or the trans-fibroreticular cell migration selectively occurs. And this is the heart. We can directly monitor the blood flow inside of the beating heart using this heart imaging chamber. And this is the data of the epicardial fat accumulation which are induced by the high-fat diet. So this white color is the lipid accumulation, which were labeled by using the special fluorophore, which is SF44. So we can acquire the in vivo imaging of the heart, and then we can also analyze this cardiac fat deposition in the heart after the high-fat diet. For the liver imaging, we slightly incise the abdominal skin and the peristome to access the liver tissue and gently take the organ tissue slightly out of the body, mounting the liver and with the cover slip. And then this is the research example of the nanoparticle drug delivery and accumulation in the liver sinusoid, which was captured by the real-time video. The red nanoparticle drugs were delivered in the liver sinusoid right after injection. So this is the right before injection, and right after injection, then we can see some accumulational dynamics to the specific sites of the liver sinusoid a long time. And then when we apply it to the fatty liver disease model, we can clearly visualize the fibrogenic collagen deposition and the lipid drumlets in the liver tissue. Especially fibrogenic collagen can be visualized with the label-free second harmonic generation signals from the two-photon microscopy system. Also, here's the imaging result of monitoring the development of the hepatic lipid drumlet in the net-filled mouse model, visualizing the disease development over time. The number of the lipid drumlets accumulated in the hepatocytes increased over the 3 weeks in the liver disease model. And we've also performed the quantitative analysis of the lipid drumlet size and area located near the various vessel type, such as the portal vein, periportal, and the sinusoidal vessels. So in the result, the size and the area of the lipid drumlets increases from the portal vein and to the sinusoidal area a long time. And in the steatosis disease model, which was induced with the 27 weeks of the high-fat diet, the hepatic lipid drumlet and also the high fibrogenic collagen were accumulated in the hepatic tissue. Especially significant fibrogenic collagen deposition in the liver stroma, which means the liver fibrosis, was successfully visualized by the label-free second harmonic generation signal imaging. Compared to the sham control model, the hepatic lipid drumlets and also the perisinusoidal collagen were significantly observed around the hepatocytes in the liver of the steatosis animal model. And also in the magnified 3D rendered images, the distinctive feature of advanced net-filled models, such as accumulation of the lipid drumlets, and also perisinusoidal deposition of the fibrogenic collagen were clearly identifiable. And furthermore, we could precisely quantify the volumetric proportion occupied by the hepatic lipid drumlet and the collagen fibers in the liver tissue in vivo. So this is another research application of the delivery monitoring in the small intestine. So here, we visualize the real-time video of the intestinal contraction in the intestinal villi together with the observed fatty acids in the lymphatic vessel. The imaging result supports that the lactea can also contract to drive the fluid within, and also the lacteal contraction itself can facilitate the efficient drainage of the fatty acids. So here we could visualize the red fatty acids can be drained to the lymphatics over the minutes after applying the materials to the intestinal lumen, and thereby we can validate the absorption and also the drug delivery efficiency of the target material to the intestinal villi over time. And this is the example of the in vivo bone marrow imaging in the cranial bone, visualizing the long-term cell dynamics of the early engraftment of the transplanted bone marrow cell in the host cranial bone marrow. So over the days after bone marrow transplantation, we can visualize and analyze that the red transplanted bone marrow cells can engraft to the specific sites and forming the cell cluster for the proliferation and differentiation in vivo in the host cranial bone marrow. Also, we can visualize and analyze some cell division and migration efficiency of the transplanted hematopoietic stem cell in the cell cluster in a quantitative manner. And for the long-term monitoring of the target organ in the normal or the diseased animal model, we can utilize the imaging window chambers such as dorsal skin fold chamber for in vivo research of the cancer or the drug candidates. And for the brain research, we can implant the cranial imaging window in the skull to visualize the brain cortex in a long-term manner. And the abdominal imaging window is just for the abdominal internal organs, including some pancreas, spleen, kidney, colon, etc. For in vivo lung imaging, we utilize the suction-based lung imaging window chamber. So we've succeeded in vivo RBC trafficking and also the measurement of the red blood cell flow rate inside of the live lung tissue. And for the long-term memory tissue and the pancreas imaging, we can utilize the memory imaging window chamber and the pancreas imaging window chamber, all of which are fully optimized and provided as the final accessory products from the IVIM technology. With an imaging technology utilizing dorsal skin fold chamber, we can perform the repetitive in vivo imaging of the tumor development or the in vivo analysis of the target drug delivery in the tumor tissue. So firstly, we implant the tumor cell, the green fluorescent expressing tumor cell, and then we monitor the development of the tumor on the site. And the result shows the tumor growth with the newly generated the vascular angiogenesis within 4 days after tumor implantation. And for in vivo drug testing, we've generated 2 types of tumor mouse models with the dorsal skin fold chamber. The one is the anti-angiogenic drug treated model, and the other one is the treated with just cell line, which is the sham control model. And by visualizing the tumor cell together with the tumor blood vessel, we can analyze the drugs anti-angiogenic effects on the tumor tissue. And the result shows that the tumor vessel can be less dilated in the drug-treated group compared to the sham control model. In the white area, after drug treatment, the vessel morphology become more normalized, that is less dilated and less branched compared to the control model. So in this way, we can finally validate some in vivo efficacy of the anti-angiogenic drug candidates on the tumor vessel. And this is another example investigating the nanoparticle drug delivery to the breast cancer cell. The GFP expressing breast cancer cell were implanted and grown for days for the drug testing. Then we treated the red fluorescent nanoparticle drug to analyze the drug delivery and the drug distribution to the target breast cancer. And over the time after the particle injection, we could monitor the delivery rate of the nanoparticle to the tumor tissue. So within 24 hours, nanoparticles are fully delivered to the breast cancer tissue like this. And for in vivo lung imaging, as you already know, when we make an incision on the rib, then the air pressure will just change, thereby the mouse would be expired. So with the in vivo optimized stabilized lung imaging chamber, we need to make the mouse breathe again by the intubation, which is connected to the ventilator. Then together with the lung imaging window chamber for the stable visualization, then we can acquire the high quality image of the neutrophil dynamics in the pulmonary vasculature. So utilizing this in vivo lung imaging platform with the stabilized lung window chamber, we can acquire this kind of high quality real-time microcirculation movie of red blood cell flows inside of the pulmonary vasculature. So this is one of in vivo lung imaging examples acquired from the healthy mouse model. So we've acquired the real-time video of the pulmonary microcirculation in 30 frames per second. Then we processed for the frame averaging and motion compensation for the vasculature. And then we do the video overlay with the RBC signal. So through this setting, we can get the high quality real-time movie of the pulmonary microcirculation and can also measure the red blood cell flow rate with the cell trafficking functions as well. And the disease model after sepsis induction by injecting LPS, the pulmonary neutrophils can migrate to the specific sites and block the microcirculation of the lung blood vessels. And it finally can acquire the real-time quantitative analysis of the blood flow, distinguish the functional capillaries, and analyze the vascular area specific velocity in the pulmonary microcirculation in the sepsis animal model. And for the long-term imaging of the mouse brain, we utilize the cranial imaging window to visualize the cerebral cortex. So it can be applied to the brain tumor imaging research. So we've implanted the glioma cell to the brain and monitor the tumor development in the same brain model, same brain imaging area in the same mouse model over the 15 days. So the result shows that the number of the glioma cells just increased with the round cell shape over the days of the tumor growth. And this is the research example monitoring the brain neuroinflammatory responses, which was induced by the 3MP chemical injection. After induction, lysin positive leukocytes are getting densely located to the specific sites of the cerebral cortex within 10 days after inflammatory induction. And also from the time-lapse imaging, we could visualize and analyze the meandering and crawling behavior of the neutrophil inside of the brain of the neuroinflammation mouse model. Then we longitudinally observed the BBB integrity through a 30-day of the neuroinflammation status. The vascular leakage in the cerebral cortex was observed in the 3rd week, and then followed by severe inflammatory responses with massive leukocytes infiltration at day 30, which is reflecting on acute BBB impairment. And for abdominal organs, we utilize abdominal imaging window for the long-term in vivo monitoring of the internal organs, such as pancreas, spleen, and the kidney. So we could visualize the real-time cell dynamics of the monocytes and the dendritic cell in the spleen like this. And also we can monitor the pancreatic islet cell using some MIP-GFP transgenic mouse. And the cell dynamics and the development can be monitored in a long-term basis by utilizing those abdominal imaging window system. And especially in the kidney, we can do the real-time monitoring of the in vivo cell behavior of the neutrophil in the kidney. So from the real-time movie data, we can quantitative analyze the cell velocity of each individual immune cell in real time. In the kidney, after islet transplantation, we monitor the islet cell engraftment and the cell development in the kidney in the long-term manner. In addition, in the colorectal cancer model, we track the development of the cancer tissue in the cecum and also the changes in the microenvironment according to the treatment of the anti-cancer drug in the same mouse model, together with the vascular changes. So again, I'd like to emphasize that intravital imaging can be a great method to monitor the temporal dynamics of the disease development in the same animal model over time. And then this is one representative data for pancreas imaging using the pancreatic imaging window chamber. So we've implanted the pancreatic cancer cell, which is PANC2 GFP cell. Then we can monitor the active proliferation and also proliferation of the pancreatic cancer cell together with the vascular structure remodeling as well. Well, we can actually monitor the every day from day 9 to 14. So we can see the dramatic proliferation of the cancer cell and also the blood vessel changes over time. So finally, IVIM technology has a wide range experimental in vivo research application throughout almost all organs in the live animal model without any restriction of the imaging targets. And then to summarize, IVIM technology is a total solution provider of the intravital microscopy technology. So we are providing the unique all-in-one intravital microscopy with strong applicational support and the high quality in vivo imaging research services for academia researchers in various biomedical research fields as well. And we have lots of in vivo research publications by using our IVIM system, which are categorized by imaging organs and the imaging purpose, such as blood cell flow analysis and such as like in vivo validation of the drug delivery and the cell delivery. Thereby, we can fully support the researchers with the optimized in vivo protocols and with the strong academic backgrounds. For the last, we've been doing active promotion and the setting for the reference sites worldwide. So we are always happy to meet the researchers in the worldwide who have interested in the in vivo imaging. So if you want to see and utilize our IVIM system to your research, please don't hesitate to contact us. So here are our customers worldwide, which includes Harvard Medical School in Boston, US, and Max Planck Institute in Germany, and University of Oxford and Imperial College of London in UK, and University of Ottawa in Canada, etc. So we are actively providing the academic support with in vivo imaging platforms and also providing in vivo research services with the optimized in vivo protocols. So that just covers about everything I'd like to share today. So if you have any question according to in vivo application, please feel free to ask them in the Q&A session. Thanks for your attention and thanks for having me today. Thank you.
Thank you very much for the great presentation. We will now take some time for questions. And the first question is, how many days can I observe the target tissue in living body when I adapt the imaging window?
Thank you for the question, Cobra. Yeah, generally, we can follow up the model as long as the window is well maintained. We can monitor it over the weeks and over the months until the end of imaging date. So for example, in case of the cranial imaging window, we can visualize the brain of the same mouse model over the weeks, over the months, and even over the 1 year. So to maintain the mouse model in a good condition, The surgical procedure should be well optimized for the best in vivo performance. So thereby we can supporting you with the training program, including some surgical procedures with the optimized material for in vivo imaging of the various target organ and the various target tissue in the live animal.
Okay, and the second question is, are there any side effects or unexpected events caused by window surgery?
Yes, actually, as window surgery includes the skin incision, thereby there's some inflammatory responses can occur if we don't treat any drugs. So thereby, in our standard protocols, we recommend to treat anti-inflammatory drugs through the IP injection of the remodel and through the drinking water from the day we did surgery and to the day right before the imaging experiment.
Thank you for the answer. And there is another question. Can I use a rat instead of a mouse?
Sure. Our system is not fully optimized only for mouse, but also we can utilize to the rat model as well. So for the rat model, we are providing some customized imaging system which are fully optimized for the rat, such as free space system for the older rat, but still the young rat can be visualized using our standard IVIM system.
Thank you for the answer. And there was another question previously asked before the webinar. How deep can we image in vivo inside the brain?
Actually, it depends on imaging target organ with different light scattering properties, such as fat and lipid and the membrane and etc. And also it varies upon the microscopy system itself. But in terms of brain, we can visualize in vivo up to 150 micrometer with confocal system. And also with the two-photon system, we can visualize up to 300 micrometer in depth. And to overcome this limitation of the imaging depth, we can utilize some ex vivo optical clearing technique for spatial information of the biological object inside of the brain tissue. So utilizing the in vivo imaging and the ex vivo imaging technology together can be a great method for the better understanding of the spatio-temporal dynamics of the various cell and biomolecules in the brain. Yeah.
Thank you for your answer. It looks like we have covered all the bases and there are no more questions. So the webinar will be ended here and it will be available on demand on our YouTube channel, IVIM Technology. As IVIM Technology, we offer hands-on and virtual demonstrations, pre-imaging tests, experiments with your samples, and training for animal surgery models. For any inquiries, please contact us at the email information@ivimtech.com. Thanks again for joining us today, and we will see you all next time around. Thank you, Selina.
Thank you, Kubra.